Dear colleagues, it's our honor and pleasure to start the second wave of the Ain Shams Neuro Academy. This is the beginning of the second waves of multiple sclerosis. We had 3 waves before of stroke, and I hope you get a lot of benefits from these expert speakers in this area. And now this is the beginning of the second subject, which is multiple sclerosis, and we are very honored and pleased to have with us one of the eminent stars of multiple sclerosis, Professor Hans-Peter Harting. He is from Düsseldorf, and also with moderator with me is my dear colleague, Professor Dr. Magd, the president of the Egyptian Society of Multiple Sclerosis. I guess we will have a very important lecture and very rich with a lot of knowledge from one of the experts in the area. So I will let my dear colleague, Dr. Magd, to present Professor Hans.
Thank you, dear Dr. Hani. And of course, we are very honored to have with us today Professor Hans-Peter Harting. As Dr. Hani said, he is one of the stars of multiple sclerosis, and we all read his publications all the time. It's a great honor to have him in virtual, I would say virtual. Maybe next time we have him in personal in Egypt when things open up. And Professor Dr. Hans is the chair of the Department of Neurology, director of the Center for Neurology and Neuropsychiatry, Heinrich Heine University, Düsseldorf, Germany, and the medical director of the Department of Conservative Medicine, Düsseldorf University Hospital in Germany also. Professor Peter has, of course, been the president of the ECTRIMS before, and has served, among others, in the executive board of the European Charcot Foundation. The CV is a very long list, so we all know Professor Harting very well. And I can see from the slides that maybe there is a new title here, the Honorary Professor, University of Sydney, Australia. So we are honored to have you with us, and we will all enjoy your presentation, and I expect a lot of questions. So bear with us these questions, and we are welcoming your presentation on the immunopathogenesis of MS. Professor Hans.
Masai Noor, thank you very much, Professor Mark, Professor Hani, dear colleagues. It is my pleasure and honor to be part of your program. As I mentioned before the webcast started, I would have loved to be in Cairo in person, but those are the days, and I hope that I will have the opportunity perhaps next year to visit your great country. So these are my disclosures. What I'd like to do this evening is put this in the context of what is the disease and what is its cause, what is the pathology of MS, which are known etiological factors that contribute to the causation, susceptibility, and severity of the disease. I will then briefly review what we know about the role of T cells in orchestrating the aberrant pathogenic immune response, then sketch out the emerging fundamental role of B cells, in actual fact, a sort of rediscovery of their fundamental importance, and then end on the question whether we are dealing in MS with a continuum of underlying pathobiology, which may bear on the therapeutic approach. You all know that the majority of MS patients start with a relapsing remitting course, followed by the transition to SPMS after a mean of 10 years or so, that for a period is associated with inflammatory activity that clinically manifests as relapses. And then there is, of course, as the disease advances, SPMS without relapses. And you also know that some 15% or so of patients have primary progressive onset of MS, an inexorable accumulation of disability. Now we've got to know over the years, and I will come back to this, that probably this strict clinical definition or distinction of the causes of MS may not be reflected in a pathobiology that encompasses both inflammation and neurodegeneration from the very beginning and throughout the course of the disease. So if one looks at MS, and you all know the clinical manifestations, the CSF findings, the MR alterations in the brain and the spinal cord, we still think that the cardinal features are the perivascular inflammation, demyelination, you see this area devoid of myelin sheath, and axonal degeneration and demise with transaction. Some 20 years ago, neuroimmunopathologists Hans Lassmann, Claudia Lucchinetti, and Wolfgang Brück try to categorize the immunopathology further into 4 patterns dependent on the predominance of T cells or B cells and antibodies, of a kind of intrinsic oligodendrogliopathy and a primary oligodendrocyte degeneration. I think it has taught us a lot and structured our thinking, but whether this is true in MS in this strict fashion is still out, I think, or the jury is still out on this. An important observation, of course, is that as the disease advances, inflammation tends to diminish and eventually abates. Initially, there is the capacity for both functional and structural recovery or regeneration, remyelination. But this, again, is sort of exhausted over time, and then degeneration, neurodegeneration takes precedence. We learned over the past, again, 10 to 20 years that unlike the classical textbook knowledge, MS is not just a disease of the white matter. It involves the gray matter, the cortex, very importantly for the clinical expression, for example, the cognitive defects that have for long been neglected. So I think the feature has become more complicated, added more levels of complexity, and even what was referred to as the normal appearing white matter is clearly diffusely diseased in multiple sclerosis. This is from the Vienna group of Hans Lassmann. They have a wonderful collection of archival material, autopsy material, histopathological material of the classical acute Marburg type MS, relapsing, remitting, secondary progressive, and primary progressive MS. And you see the various topographical distribution of changes, the demyelinated plaques here, again across the course of the disease. Then what I mentioned already, the cortical demyelination, both in primary and secondary progressive MS, deep gray matter demyelination, and these dots which are reflective of inflammatory infiltrates. So MS may, and it still starts as a focal inflammatory disease of the central nervous system, primarily in the white matter, but with chronicity, with advancement of the disease, inflammation becomes diffuse and is associated with progressive axonal injury in the normal appearing white matter and cortical demyelination. You've seen such a cartoon in multiple ways before. It sort of captures the subclinical, clinical disease, the MR equivalent of inflammatory activity, much more frequent, of course, as we know, than clinical relapses, the extent of inflammation predominant during the earlier stages of the disease, and at the same time, neuroaxonal loss giving rise in accumulation to a loss of brain volume, as indicated here. We have now the radiologically isolated syndrome, where patients sort of incidentally upon diagnostic workup are found to have on their MR brain scans lesions compatible with inflammatory demyelination. Then the clinically isolated syndrome, the first clinical manifestation, and then the forms that I described before. Of course, MS is a disease that spans decades and has severe impact and puts a very dramatic burden on the individual patient, his or her caregivers, our health systems, and society at large. Just another rendition of this prototypic evolution of multiple sclerosis, but this time also showing this prodromal phase preclinically, where during early adolescence, most likely predisposing genes also interacting with environmental lifestyle factors determine when a preclinical activity turns into a clinically manifest disease. And again, with the first stages of the disease, the 10 or 15 years of relapsing, a relapsing course, we feel that systemic adaptive or antigen specific or cognate immunity is the main driver of the pathobiological processes, whereas later on during progression, innate immunity, antigen non-specific immune responses, mainly driven in the central nervous system by microglia, takes center stage. So talking about these factors that determine disease susceptibility and to some extent also the prognosis, the severity of the disease. It's a complex disease like many autoimmune disorders, resulting, so we think, from a complicated interplay of genetic factors, particularly those that shape immune reactivity. In the meantime, GWAS analysis identified more than 250 single nucleotide polymorphisms, and environmental factors, which are, for example, infections, but also sun exposure, vitamin D levels, and then the lifestyle factors of obesity or smoking. We know that this, during adolescence or at the earliest clinical signs of multiple sclerosis, greatly impact on the risk of developing the disorder. And then we have an internal environment, an area that for many diseases has become the focus of extensive research, and that's the gut microbiome. And we will talk about this and review current hypotheses of how that can impact misguided immune responses leading to multiple sclerosis. This is a not so recent review that from the Karolinska group that looked at the odds ratios that come with lifestyle and environmental factors. And as our colleagues from Stockholm have intensively investigated, some interact with HLA or MHC genes, thereby increasing the likelihood to develop multiple sclerosis, and some don't. So EBV serology by itself, 3.6-fold increase in the risk to develop multiple sclerosis. Mononucleosis, low sun exposure, vitamin D deficiency, smoking, obesity during adolescence, and some other factors are associated with an increased risk to develop MS. And when you compare these odds ratios in combination with interactions of certain genetic makeup in the HLA regions, these likelihoods get even higher. Let's have a look, for example, at smoking. There are many studies now on thousands of patients who have unequivocally established the increased risk of smokers to come down with multiple sclerosis. But again, interestingly, the point is there are specific immunogenetic setups, the correlation with HLA-DRB1, that interact to further augment the risk to develop multiple sclerosis. Now, how does this work? There are several hypotheses based on multiple observations, for example, of the effect of smoke components to change methylation, to cause epigenetic gene dysregulation that also impacts on the way the immune system works, sort of brings down the number of regulatory T cells. Very importantly, damages or disrupts the blood-brain barrier. And also components of smoke have direct effects on microglia. They promote iron accumulation in myelin and oligodendrocytes. And then T cells are licensed in the lung, in the inflamed lung, to then enter the central nervous system and set there in motion an inflammatory cascade that eventuates damage to the myelin sheath and to the neurons and axons. Let's now turn to sun exposure and its role in causing or conveying susceptibility to multiple sclerosis or increasing the risk. And this is from the group of Alberto Askerio at Harvard, where they looked at patients with self-reported outdoor exposure and looked at the relative risk to develop multiple sclerosis. And you see it's pretty clear that with high exposure, the risk is lower. The risk seems to be lower if there is high exposure to sunlight during summer, but also during winter if you compare low with high. And they also looked again, examining and studying questionnaires, the direct sun exposure, and find that a low exposure comes with a higher risk to develop multiple sclerosis. Now, as you of course know, there is a clear correlation between the generation of vitamin D and the exposure to sunlight. This is a study that summarized, or a review that summarized findings to relate the levels of vitamin D or the extent of vitamin D deficiency with disease activity. What you see here is that with lowest levels, the risk is highest compared to high levels of vitamin D, or the metabolite 25-hydroxy D2. And again, if one looks at a quintile, it's clear if you are in the lower quintile, you have a higher number of inflammatory activity as gleaned through cerebral MR. Vitamin D as an early predictor of MS activity and progression in the benefit trial cohort. You will remember the benefit trial was a trial in clinically isolated syndrome where interferon beta 1b, or placebo until they developed clinically definite multiple sclerosis. The trial was initially blinded and controlled for 2 years, but there was an extension which goes on now in actual fact into the 17th year from the beginning. What you see here, the probability of clinically definite multiple sclerosis after 1 year is significantly higher in patients with low vitamin D levels compared to those with high vitamin D levels. The cumulative number of new active lesions up to year 5 is higher in patients with low-level vitamin D. Similarly, T2 volume higher in vitamin-deficient patients, and brain volume loss is more rapid in patients with low vitamin D. So pretty consistent data from this very interesting patient cohort demonstrating a role of vitamin D as a predictor progression. Let's now turn to infections, and I think the one agent that has stood the test of time as being most likely of causative import is Epstein-Barr virus. This is again a study from the Harvard group of Alberto Asquerio, Cassandra Munger, who summarized in this review the risk in patients with EBV positivity, serological positivity, in the absence of a history of mononucleosis in early childhood. This is set as the relative value in terms of the risk as 1, and look and compare this. The few patients, I have to say, that are EBV negative have a much lower risk. The risk is higher, is more than doubled if there is a history of mononucleosis. What are the ideas we entertain about how EBV is associated with MS, or I should say, with a range of other autoimmune diseases? Well, the propensity of EBV to infect naive B cells appears to be of great significance. And I will bridge this with the current thinking about the important role of B cells later in my talk. So you have EBV invading naive B cells. They are then becoming activated B cell blasts or plasmablasts, expand in the germinal center of a lymph node, and then give rise to memory B cells. And we know memory B cells are increased in number in multiple sclerosis, and they turn on B cells and cause enhanced interaction with T cells, and also increase the manufacture of autoantibodies. Corroborative evidence came from this Stanford group that looked at immunohistology of patients with chronic active disease, chronic progressive disease compared to controls, and could in fact detect a number of latent and early lytic proteins, components of EBV in MS. Interestingly, the group in Rochester, Buffalo, could associate a humoral immune response to EBV components with the development of cortical atrophy and lesion burden in multiple sclerosis. So I think the story is still, or the evidence in aggregate, circumstantial, but I think amongst the many infective agents that have been associated with MS causation, EBV still remains the most likely candidate. This brings me, however, to something which has emerged over the past years and which has caught our interest in Düsseldorf, but with a group in Lyon, and that relates to the role of human endogenous retroviruses in multiple sclerosis, but also in other diseases. You know that 8% of our genome encodes for endogenous retroviruses. And the idea is that during revolution, primates somehow acquired this. This genetic material is sort of silent over long periods of time, but then may become activated, may be expressed, and then give rise to a range of responses that I will describe in more detail. This is a study from Amsterdam, the Dutch Brain Bank, where human endogenous retrovirus W, or the so-called envelope protein, could be identified in MS lesions. This is the brownish color that you see here. Strong in and around the vessels. Our group, David Kramer, in collaboration with our colleagues in Amsterdam and Lyon, found that obviously this retroviral envelope protein inhibits oligodendroglial precursor cells identified by the marker PDGFR alpha. One finds microglia expressing TLR4, toll-like receptor 4, associating with oligodendrocyte precursors, and the presence of the envelope protein is associated with a reduced number of the precursors of oligodendrocytes. And if one looks in vitro or in vitro at the differentiation of OPCs into fully functional remyelinating oligodendrocytes, one can block this by the human endogenous retrovirus envelope protein. And as we have also shown that the animal model EAE, or in the animal model EAE, if you administer a neutralizing antibody against this protein, you can enhance remyelination. More recently, we looked at the role of this protein in activating microglia and causing microglial-mediated axonal degeneration. You see here the envelope protein associated with microglia expressing MHC class 2, but also the toll-like receptor 4. And here we looked at axons and see that the envelope protein expressed by microglia is in very close proximity to degenerating axons. So we feel that this is another mechanism by which this particular envelope protein participates in the pathogenesis of multiple sclerosis. In parenthesis, there is an emerging body of evidence to implicate a different endogenous retrovirus in motor neuron disease, or amyotrophic lateral sclerosis. This is, before I sort of escape this, this is from the same paper, a study in vitro of the impact of this envelope protein on the expression by microglia of regenerative factors. These are the controls. This is microglial cells exposed to the retroviral envelope protein, and we determined in the supernatants regenerative or axon recovery promoting or axon integrity and survival promoting factors, IGF, insulin growth factor 1, CSF1, fibroblast growth factor 2. So you see there are apparently different ways by which microglia in the presence of this retroviral protein causes neurodegeneration and diminishes repair. So this is to put this into a context in cartoon fashion. I just want to emphasize again, and I didn't mention that the endogenous proteins obviously need to be activated in the genome, and there is a clear correlation with herpes virus infection. So HERV-W is activated, the envelope protein gets secreted, acts upon microglial cells, renders them angry to generate and release cytokines. This in turn then recruits other T cells. But importantly, there is also the role of this protein in blocking the differentiation of oligodendrocyte precursor cells into remyelinating oligodendrocytes. And the protein is located at the surface of microglial cells. Now, the translation was done in a couple of phase 1, phase 2 trials, and I just want to briefly introduce this ANGEL trial, the Change MS ANGEL trial, where in a double-blind randomized placebo-controlled fashion, different doses of this antibody that neutralizes the retroviral envelope protein were administered to patients at 4 weekly intervals for an initial period of 6 doses, so half a year, when placebo patients were switched to receive either one of these or one of these doses. Now, this was a population of relapsing remitting MS patients, and the primary outcome, of course, as typical of such phase 2 trials, was inflammatory activity as gleaned through MRI. So unfortunately, and we hope that we get the paper published soon in the Green Journal, unfortunately, the primary outcome for the protocol defined patients and regimens was not reached. However, we have the feeling that with higher doses and longer exposure, there may be an impact also on inflammation. But very importantly, we noted, and this I think is surprising over this period of time, a significant effect of the neutralizing antibody on the evolution of either whole brain or regional atrophy. You see here the relatively clear and rapid volume loss in the thalamus with placebo, and a kind of dose-dependent diminution of thalamic atrophy with the monoclonal antibody. Similarly, there is an effect of the higher dose of the monoclonal antibody in slowing cortical volume loss and consistently whole brain volume loss. Another part of the study was to look at MTR bands in proximity to the ventricles, which have been considered a reflection of myelin integrity. And again, this antibody temelimab apparently sort of maintained myelin integrity to a higher extent compared to placebo. So another trial, now a combination trial, is about to get started at the Karolinska group, and we hope to also look in the future at patients with progressive disease. Now, coming back to what I refer to as the internal environment and the role of the gut microbiome. You know, this is an area that has become pretty hot with relation to multiple neurological disorders, Alzheimer's disease, Parkinson's, many other non-CNS autoimmune diseases. You all know that we harbor, I would say, tons of different strains of bacteria, and these have been correlated with disease activity. We know that the gut contains not only a very large kind of nervous system, neurons, but also an entire set of immunological factors, cellular, humoral, that may be very important upon initial sensing of external agents that are in the gut and shaping the repertoire of cells. And very interestingly, and I will tell you this in a moment, these T cells from the gut-blood interface can be rendered activated and governed to travel to the central nervous system. There was an early paper from the Max Planck Institute in Munich looking at the effect of commensal microbiota on myelin autoantigen-driven EAE. And they compared the development of spontaneous EAE in germ-free conditions, so no microbes present, almost zero clinical expression and pathological expression. However, when and this is in contrast the pathogen exposed group, and when one colonized these germ-free animals with microbes, they also developed EAE. Now, many groups looked at different compositions of the microbial habitat in MS. And not unexpectedly, there is no uniform picture emerging. You know, the different geographies, genetic influences, dietary behavior clearly are significant in the composition of the bacterial habitat in the respective microbiome. But this is a review that looked at the relative composition or prevalence of certain strains and species, bacterial species. A number of them found decreased, others increased in relation to controls. But to date, no convincing, clear-cut picture, that's my reading, has emerged. But it's still an area of great scientific interest, and this paper caught the attention of the neuroimmunologists because it comes back to an old concept, how T cells are, or autoreactive T cells are activated in multiple sclerosis, the concept of molecular mimicry. So this is the most recent and very stringent evidence produced by the colleagues in Zurich, who found this specific antigen, bacterial antigen, with its immunodominant epitopes being able to stimulate CD4 T cell specific neuroantigens in patients with a predisposing immunogenetic makeup. And this very readable editorial that came with the paper sort of illustrates again that there is the possibility that bacteria encounter antigen-presenting cells, and there is, by chance and bad luck of nature, structural homology with a myelin component, so that T cells recognizing these bacteria, or that these bacterial peptides, through their molecular similarity or identity to myelin proteins, can activate myelin reactive CD4 cells to travel to the central nervous system. I would now like to turn to a sort of deeper analysis of the role of T cells and B cells in multiple sclerosis. As you know, T cells have for long been considered the main drivers, almost the exclusive drivers of the pathology of multiple sclerosis. And this was driven initially by two lines of evidence: immunopathology first, that there is a significant perivascular, but also more distant accumulation of T cells and a predominance of cytotoxic CD8 cells compared to CD4 cells, and the existence of the T cell dependent experimental models of EAE. Also, one can retrieve activated autoimmune T cells from blood and central and cerebrospinal fluid, and also, if one is lucky enough to find biopsies and analyze them from lesions. More recently, the genome-wide association studies emphasized the role of genes that govern T cell immune responses. And as I mentioned, not just in this order, but the importance of T cells shown to be essential prerequisites for animals to develop EAE spontaneously or upon transfer of autoreactive T cells and T cell lines. Sergio Baranzini and Jorge Oxenberg have nicely sort of reviewed and commented on these genomic findings. I should say there is also evidence that genes encoding neuronal components, nervous tissue components, are differentially exposed in MS patients. But let's, for the moment, and in the context of our topic, focus on those multiple genetic polymorphisms that all dictate how T cells interact with each other, which regulatory signals are emanated, acting upon macrophages, on monocytes, and determining also the relative proportion of pro-inflammatory versus regulatory T lymphocytes. But also, and again relevant in the context of our reassessment of the role of B cells, is that there are genomic regions associated with MS that are active in B cells. If one looks at the sequence of immunological events, and again, you've seen these kind of cartoons in different renditions before, we think that the immune repertoire of individuals destined to develop multiple sclerosis harbor in the lymph nodes, in the bone marrow, cells that can potentially recognize antigens in the CNS. However, they remain dormant for years or decades. It requires an activating signal or a danger pattern, as the immunologists say, to activate these dormant cells, which allows them to leave the periphery, the blood, through the blood-brain barrier, to enter and conquer, if you like, the central nervous system. Well, these cells can only survive and proliferate in the central nervous system if they interact with an antigen-presenting cell. The probably most relevant in the brain and spinal cord is the microglia. This also requires a specific microenvironment, inflammatory milieu with a number of components. And only if all these conditions are fulfilled, the invading T cell proliferates and releases cytokines, enlists the help of other cells resident or immigrated to effect damage to the myelin sheath and the axon or neuron. You already see here, but we will have to belabor this in more detail, that of course we thought that B cells are important. They manufacture and secrete antibodies, including demyelinating antibodies. Now, this all occurs in the context of a dysregulation of pro-inflammatory and immunoregulatory molecules and cellular populations. I would now like to focus on B cells. B cells are present in all clinical phenotypes of MS, but they are most numerous in relapsing, remitting, and interestingly in primary progressive multiple sclerosis. More active in elevated in numbers in active lesions, more so than in slowly expanding or inactive, and particularly enriched in the meninges. And you will remember the intriguing paper from the Rome group published in 2006, Magliozzi and colleagues, who described aggregates of B cells in the subpial region of patients with SPMS and related the presence and number of these B cell aggregates with disability and disability progression. So the more B cell aggregates could be found, the faster was the accumulation of disability in these patients. We know with further pathological scrutiny that it's not just B cells. You also see T cells, CD4 or CD8 in these areas, which are now referred to as a compartmentalized site of compartmentalized inflammation. You can also visualize this in vivo with high field strength MRI, as very nicely shown by Absinter and colleagues. And it has been postulated that these sites of compartmentalized inflammation are crucial for the pathobiology in the progressive stage of the disease. So they represent, if you like, a mini immune system in situ. There is evidence that these ectopic lymphoid tissue, that's another technical term to describe this, has all the components or many components of the immune system and can drive and maintain an inflammatory response during progression. Well, the true success in translation and the ex posteriori proof that B cells are very important comes from the result of the clinical trials looking at the effects of B cell depletion. CD20 B cells depleted with rituximab. That was the first paper we published in 2005. A couple of patients, there were another series of cases, Bruce Cree, and then Steve Hauser embarked on this phase 2 trial and could demonstrate that rituximab in the typical regime, initial induction, and then later on in extension every 6 months, produced a precipitous decrease in gut-enhancing lesions on MRI. So within a matter of 12 weeks, the number of T1 gadolinium-enhancing lesions was very low. Compare this with placebo patients. They sort of accrued these lesions over the 6 months of this phase 2 trial. Now, rituximab was not further developed for a number of reasons. There was a then humanized antibody ocrelizumab, also depleting CD20 cells, that was examined in 2 trials in relapsing remitting multiple sclerosis and in 1 trial in primary progressive MS. You know the results in both populations. Ocrelizumab was highly effective in primary progressive MS in a smaller subgroup and not so markedly, but still. But again, you see from these trials, the RMS and PPMS trial, very rapid depletion to 0 levels of B cells in the periphery. And again, a further look at the relapsing twin trials where ocrelizumab was compared to interferon beta-1 AIM showed an early effect related to depletion of B cells on inflammatory activity on MRI. And looking at this, it became clear this could not be an effect on autoantibody production. This was too early. The kinetics wouldn't be compatible with the traditional view of an impact on autoantibody production. Just for completeness, the relative recently published studies of ofatumumab versus teriflunomide in relapsing remitting MS again showing superiority of this approach with a human monoclonal anti-CD20. Very impressive results with this subcutaneously administered monoclonal antibody. Steve Hauser called this the new era of therapy, B-cell therapy, initially rituximab, ocrelizumab, ofatumumab. There's another one in development, ablituximab. And as you know, they do not deplete the entire lineage of B cells, but only those cells that carry the CD20 antigens. This is important because it leaves intact stem cells, pro-B cells, for B cell reconstitution if need arises, for example, to fight off an invading foreign microorganism. And it also leaves the plasma cells, the site of manufacture of antibodies and the imprinted humoral immunological memory intact. So natural defense mechanisms, at least in theory, should be intact, and obviously this would be of importance with any kind of immuno-interventional strategy. So these studies also then showed that it is not just autoantibody production by which B cells damage the tissue in the MS. It's not just the production of cytokines. The new information was obviously the presence of aggregates with high numbers of B cells, subpelially, that may be crucial in driving inflammation during progression, but very importantly, antigen presentation. There is data from animal models to show that MHC class II dependent B cell antigen presenting function towards T cells occurs or is required for the induction of EAE independent of demyelinating antibodies. So B cells provide signals to promote T cell responses. And in turn, T cells provide help to B cells also to differentiate then into antibody producing cells. So this has sort of led to a paradigm shift. The traditional view, as I mentioned earlier, being that MS is primarily governed by the T cells. They tell the B cells to release antibodies. But the sort of emerging view is that there is a bidirectional interaction between T cells and B cells, and maybe B cells are in actual fact initially more important. And of course, immunology has shown us that there are also very heterogeneous populations of B cells, both immunoregulatory and immunopromoting, and that the balance between anti-inflammatory and pro-inflammatory B cells, like that of anti-inflammatory and pro-inflammatory T cells, is disturbed in multiple sclerosis. Again, a very exciting paper from Zurich underscores this very important role of B cells and demonstrating this critical interaction between B cells and T cells. Memory B cells, and I alluded to their increase in number in multiple sclerosis earlier, appear to activate autoreactive CD4 cells that are about to home to the central nervous system. So a process called autoproliferation, again in particularly genetically disposed MS patients, causes the activation of these autoreactive T cells, and this signal is provided by memory B cells. What about effector mechanisms of axonal neuronal damage? Now, this is the sort of more final pathway, and we all know the seminal paper by Bruce Trapp showing that axons are significantly damaged or transected even, and this dictates the extent of permanent disability. And a crucial role has been ascribed to energy failure of the mitochondria secondary to oxidative stress, and a change in the demyelinated areas of electrolyte ion channel proteins, which in effect causes the activation of destructive proteolytic enzymes that then contribute to the axonal demise. Microglial activation is very important and can be visualized, as you know, for example, by TSPO PET imaging. And microglial activation as a reflection of an activated innate immune system in MS has been considered, or is considered, a fundamental driver of the chronic inflammatory progressive disease. We already mentioned the visualization of the compartmentalized immune system, and with even higher field strength, one can see very subtle changes in the cortex that can be clearly correlated with clinical progression. But, and that's important, you can observe these changes both in relapsing forms of MS and primary or progressive MS, I should say. You know that the course description has changed in recent years, and it may be legitimate to give up a strict distinction between primary and secondary progressive MS and just group it under the heading of progressive MS. Here you see slowly expanding lesions rich in iron that one can visualize, iron-laden microglia and or macrophages at the edge of lesions. Again, you see that in the majority of relapsing patients, even more so in primary progressive MS. Slowly evolving, expanding T2 lesions, again, those that undergo local concentric and constant expression, reflecting chronic inflammation seen in the majority of relapsing and progressive, primary progressive MS patients. So the question whether the pathogenesis of pathobiology can be strictly separated represents a duality, or rather a continuum that occurs, may change quantitatively, but is characteristic of the entire evolution of multiple sclerosis, is an open question. Again, let me summarize. Inflammation may cause conduction block, demyelination, and axonal injury you know, this occurs very early on. Repeated attacks may be driven by invading systemic immune cells. There is a predominance of adaptive or cognate autoantigen specific over innate immunity, and at least for some period of time, remyelination and functional compensation enlisting supplementary neuronal circuitry may compensate and mask the damage. However, with progressive disease, these remyelinative and compensatory mechanisms may be compromised or completely stopped, and the loss of axons neurons, along with the remyelination failure, the predominance of innate versus adaptive immunity, a less overt compartmentalized inflammation may contribute to the damage. And on top of this, of course, are the changes occurring with advancing age, secondary to comorbidities. They may be superimposed, even the sort of changes that attend quote unquote normal aging. So I come to my conclusions. I hope I haven't exhausted you too much. I started out describing the disease and its causes, and again, also in terms of operational classification, we have seen and are seeing revisions. The pathology of multiple sclerosis that is more widespread than initially thought, affecting both white and gray matter. I summarized the variety of etiological factors that determine the susceptibility to multiple sclerosis and at least in part may impact the prognosis. I then turned to the immunopathogenesis proper, describing the role of T cells and emphasizing what has become very clear following the B-cell depleting therapeutic trials, the comprehensive and fundamental role of B cells. And I ended up with posing the question, and in actual fact, in a way, answering them, at least my view of whether MS is a group of diseases that with very distinct causes, or whether this disease is a continuum, both in terms of clinical presentation, underlying pathobiology, and susceptibility to therapeutic intervention. So with this, I'd like to finish and thank you very much for your attention. I'm, of course, happy to answer questions.
Thank you, Professor Hans, very much for this comprehensive presentation and in-depth update for the knowledge in pathogenesis and pathophysiology of multiple sclerosis. I will start by a couple of questions regarding the stage of the innate versus adaptive immunity in the stages of the disease. So probably, as you just mentioned, as the disease, as the MS progresses, it will pass, the main player will be the innate immunity. So you think we are in a stage now of developing new drugs working on the innate immunity, or still, because, you know, of course, most of the drugs are working on the adaptive immune system?
Right. I think a very attractive class of agents that by their physicochemical properties can enter the brain easily and also may access those areas of compartmentalized inflammation are the BTK inhibitors, the Bruton tyrosine kinase inhibitors. Evobrutinib, you know, the results of the phase 2 trial, and the ongoing comprehensive phase 3 program with this particular inhibitor, but there are also the Roche and Sanofi representatives of this class. They, I think, are very interesting. However, again, one has to keep in mind that the microglia come in different phenotypes. There is, although the concept is still contentious, there is the more pro-inflammatory M1 phenotype. There's the more anti-inflammatory and remyelinating permissive M2 phenotype. So one will have to see how the balance is hit and whether this approach really then results in a significant impact on disability progression.
Okay, thank you. I have another question regarding the, it's a very interesting concept, the retrovirus embedded in the human DNA. But what is the reason for the activation of this part of the DNA, which should be silent?
Right. I didn't go into details. There is at least an association with herpes virus infection. So most likely, or that's one hypothesis for which there is some supportive evidence, is that it takes another virus, and now not an endogenous virus, to activate the dormant part of the genome. Again, there are a few studies on this. It is, in my view, an attractive concept and also candidate. So a coincidental role of viruses.
Okay, so I will give the microphone to my dear colleague, Dr. Magd, to ask the questions.
Thank you very much, Professor Hans, for this, as Dr. Hani said, in-depth details of the pathogenesis. We enjoyed the presentation very much, and we learned a lot from it. I have a question about the link between the environmentals and the genetic factors. It seems that these different environmental factors play their part in specific genetically susceptible individuals. Has there been any link between each specific environmental factor and a specific genetic phenotype, or let's say smoking in this kind of individual, or vitamin D in that kind of individual, or has there been a link between these?
Well, basically, there is only one group that has studied this in detail. That's the Karolinska group, Thomas Olsson, Jan Hillert, and they could identify certain DR alleles that would increase the risk of smokers to develop multiple sclerosis. How exactly the interaction works, is it methylation, demethylation, and so on, I think we don't know. But this group has really looked carefully and did identify, again, in the genetic area that is still the most relevant in conveying a risk, that's the DRB1 area, established a relation, not with all environmental factors, but with some.
Thank you. And I have another question about the effect of the B-cell therapies. We know that probably a large part of their action, as you mentioned, is through the T cells also, because the B cell therapies, they don't actually change the level of the oligoclonal bands in the CSF. And maybe I've heard explanations before about the long-lasting effect of the plasma cells or the plasma. But I think that there should be a connection by time, at least, that these B cell therapies should reduce the oligoclonal bands, but they don't reduce the oligoclonal bands. So do you have another explanation for this finding?
First of all, there are obviously a few studies where you have long-term sequential CSF determinations of OCBs. Again, the idea that has been entertained, and I don't have a better explanation, is that you have these long-lived plasma blasts in the bone marrow, in the tissues, that you do not deplete, at least for long. You obviously, with the B-cell depleting agent, sort of curtail the influx from the periphery into the central nervous system. But those cells of the B-cell lineage that are already homing there are most likely not affected. And I mean, it's pretty clear the immune system is as complex as the central nervous system. There's so much redundancy built in, both on the B cell side and on the T cell side, that there will be sort of quote unquote escape pathways of continually driving a pathogenic immune response.
Because the only drug that has been proved to reduce the oligoclonal bands was natalizumab. So it seems that even with the B cell follicles inside the central nervous system, then the presence of the oligoclonal bands to a degree depends on what's coming from outside.
Right, absolutely, I agree. Yeah.
I have a question also regarding the 4 pathological types originally described by Lucanetti. You think this theory is still holding, and we have different types for different phenotypes of the disease, or still it's a mix between the 4 types described, or it's now totally different?
Well, I think it was, shall we say, heuristically important to come up with this concept. I know pathologists who never subscribe to the view, and I would definitely think that the vast majority are cases where T and B cells predominant over an intrinsic metabolic oligodendroglial pathology. Consider that these results are based on still relatively few cases histopathologically and by autopsy. Can one draw these conclusions? Can one so strictly separate? I think, as I mentioned during the concluding slides, that I tend to think, and I think there's good evidence that it's probably a continuum, and the boundaries are or have become blurred over the past years.
I have one last question regarding since with the progression of multiple sclerosis, the main player, as we were just discussing, the innate immunity, and also the aging process will start to play a role in the progression of the disease. You think it's wise to start thinking of stopping the drug? I know, of course, there is a study of Crosby and other studies, but you think at that time we should think of stopping, let's say, if the patient's above 50 or something like that, and it's taking the drug for a decade or so, we should stop it, or we should go on with the drug on assumptions that it's preventing the disease?
This is a tough question at the end. You're absolutely right. I mean, there comes the issue also of immunosenescence. Does the disease kind of naturally calm down? I'm not so sure. I personally would make it really dependent on an assessment of how is the patient doing, and still weigh the risks of discontinuation against the benefits. So I would like to await the results of the trials that look at this in a rigorous manner. At the present time, age doesn't sort of dictate me to stop the treatment. Well, you may, if patients are 70, 75, you may think differently, but then they also have comorbidities that are per se perhaps relative contraindications to therapeutic intervention. But I'm a little bit afraid and maybe cautious in considering to discontinue.
I think for this point, it's a very important point, because we have two categories of patients in the older age group: those who have been taking medication for a long time and have been stable, and maybe this is the discussion you've been talking about. And then those who develop the disease at a later age. We see some of these cases that they start having MS activity at the stage where they are also going into the immunosenescence. So maybe the second category of patients, would you have certain selections? I know this is a little bit out of the scope of your lecture, but would you have, it's an important clinical question we always face, would you prefer certain selection of medications in these age groups that start their activity at a later stage for fear of the immunosenescence and the comorbidities? And
very important, very difficult question. I think that the older a patient is or presents, again, you need to look at a computation of prognostic factors. Is he or she going on rapidly or just very slowly? Obviously, if rapidly, I would still use high efficacy drugs. I mean, like B-cell depleting agents, even if I know that the immune responses may not be as vigorous. But again, there is a range of severities that one encounters in the older age group. And I would probably gauge it according to how you see the patient develop over the next couple of years or so.
Thank you.
I don't have any more questions. I don't know whether Dr. Magdi is having any questions.
We would like to talk to Professor Peter all night, but I think we've exhausted him enough. So I will stop at this stage then. Thank you. Thank you, sir.
Thank you very much. Thank you very much. It was a pleasure seeing you from the distance.
Yeah, thank you. And we hope next time we will see you face to face in Cairo.
That would be wonderful.
A big event is the opening of the new Egyptian Museum. So
the new mummies, right? Also displayed there. Yeah, I saw it on TV. That's fantastic.
We're waiting for you.
Very good. Stay safe. Thank you so much, and good night. Bye-bye.